everyone. Welcome to episode 20 of the Powerhouse Podcast, where we bring to you the powerhouses of the mitochondrial disease world. I'm your host, Brian Harmon, President and CEO of the United Mitochondrial Disease Foundation, joined as always by my trusty co-host, Dr. Phil Yeski. Phil, it's almost hard to believe we're at 20 episodes.
Yes, it is. As soon as you said that, I started thinking, wow, I guess the time goes by quickly.
Well, don't count on our YouTube playlist where you can see all of these, because I'm almost certain I messed up the numbering somewhere. But we're going to call it 20 today. And a very special episode. We'll get to our guest here in a little bit. But of course, some exciting things, as always, happening here at UMDF as we close a calendar year. It's always a time for us to reflect and celebrate and, more importantly, plan for 2023. And one of the big milestones that we just recently hit was our 1,000th registrant in our MitoShare patient registry. And we can't say it enough how important it is to fill up that patient registry.
Can't say it enough, often enough, loud enough. It's really critical to the success that we all hope to have in terms of being able to bring therapeutics to market that are meaningful for our patients. And so it all begins with identifying the patients, characterizing the patients, engaging that patient community so that they can be involved in clinical research. So huge thank you from my side to everyone that has entrusted us in this endeavor by signing up for MitoShare. For those of you who haven't as of yet, who may be watching this, please consider it. Reach out if you have any questions. 1,000 is just the start, Brian.
That's
as
right.
You know, right? We feel, and I think credibly, there are 5,000 or 10,000 people out there that we could be engaging with. We just have to improve our reach, and we have to improve the diagnosis of mitochondrial disease, which is really the other big initiative for 2022, isn't it?
Yeah, you talk about the magic word there is engaging, right? And so much with MitoShare is both acquiring and engaging. And we're going to be talking a little bit with our guest today about how we are activating MitoShare to advance some engagement opportunities. But again, as we think about that complete picture of making sure we're charting a course towards diagnosis, some exciting pieces on the genetic testing program front as well.
Yeah, so this is the clinician-facing program we launched a couple months ago. We started with the Mitochondrial Care Network. We got good response from the healthcare providers associated with the MCNs. But now we're ready to push this out to even broader audience. We know there are many healthcare providers out there that are seeing mitochondrial disease patients, may suspect their patient clinically as a mitochondrial disease, but for whatever reason have not had access to genetic testing. That's who we're targeting. We're trying to fill that gap for those that haven't had access to genetic testing, and there's a sound clinical basis for suspecting it to make sure that they can get that testing done, and hopefully that provides a diagnosis, whether it's mitochondrial disease or some other disorder. We've advanced the diagnostic journey of that patient, and that's meaningful.
And knowing that UMDF will always serve as a home for patient families, no matter where they are in their diagnostic journey. And we think a lot about that this time of year, right? Where we want to make sure that our patient family support is an absolute paramount. We know this is a time of year that can be tough for patient families and tough for all of us. But just to make sure that UMDF is staying focused on providing ample support programs.
Absolutely. And that really ties the two programs together, right? By no means is MitoShare restricted to only those who have a confirmed genetic diagnosis of mitochondrial disease. MitoShare is meant to be the home for all patients, wherever they are in the diagnostic journey. Could just be a collection of symptoms that they've self-identified with that is consistent with mitochondrial disease. It's an okay starting point, right? But hopefully, right, by continued engagement, we can help that patient progress to finding the right clinical care, advancing towards a clinical diagnosis that can then be supported by genetic diagnosis. It's a whole spectrum. All are welcome inside MitoShare, and we really do need that pipeline to be as broad as possible.
We certainly encourage our clinical community, our patient community to visit us at umdf.org to learn more about all these programs. And, something, again, I think about the programs that have launched and the big plans we have for 2023. I'm proud to be CEO of this organization. I'm equally proud to be a fundraiser for this organization. So much of what we talk about here, in the end, it's about that bottom line of what we can do to raise funds to help power these programs. And this, for our organization, is a very special time of year as we launch our Give Energy Giving program. And I just want to give a special shout out already to legions of supporters and donors who have said mitochondrial disease patients and the families that UMDF serves is a priority for our philanthropic giving. And as we continue to work through this year end, our hearts continue to turn to our donor community who really are at the grassroots level of moving these programs forward.
It's another case where we can't say it often enough or loud enough. None of this happens without our donors. And, as someone responsible for the research mission within UMDF, I'm so deeply appreciative of all who entrust UMDF with their dollars to help us advance our mission. It's what makes the engine go.
And selfishly, it helps me plug a little fundraiser I'm doing, Phil. So as part of my New Year's resolutions and under our Activate brand, so activate your mitochondria. As we know, there's a lot of special importance to putting your mitochondria to work as it powers our bodies. I'm taking an undertaking of doing 5,000 push-ups through the month of January to do some fundraising and awareness around that in a month. I spent 160
for the whole year.
Oh, no, that's the month of January. And I signed you up too, Phil. You can join our team at umdf.org. And if push-ups aren't for you, right? And if your goal this year is to walk a little bit farther, right? Your goal is to have a different outlook on your day every single day. Whatever that means for you and your resolutions, there's a way that we can kind of all activate our spirit and our mitochondria to do good for UMDF and our patient families.
Yeah, it's a great idea, Brian. And, I'm happy to participate. I know I'm going to be sore and in pain, but a little bit of that is maybe it's walking in the shoes of what our mitochondrial disease patients go through so often on a daily basis. And it's a reminder, right, of the challenge of having a disease like mitochondrial disease. And we'll do what we can to try raise some money around that, and short-term pain, long-term gain, right?
Absolutely. Well, I know our special guest today too is no stranger to activating her mitochondria in many different ways, and we'll talk to her about that. I'm really delighted to welcome into the Powerhouse Podcast today, Dr. Marni Falk. Dr. Falk is a clinical geneticist who serves as executive director of the Mitochondrial Medicine Frontier Program. Her translational research lab investigates the causes and global metabolic consequences of mitochondrial disease, as well as targeted therapies in C. elegans, zebrafish, mouse, human tissue models of genetic-based respiratory chain dysfunction. I've had a tour of Marni's lab. I got to see the zebrafish, but we're going to dive a little bit more deeper into that and just get to know Dr. Falk today. Marni, thanks so much for joining us today.
Hi, Marni.
Thank you. Thank you both for having me.
Well, it's an exciting time here at UMDF. It's an exciting time in the mitochondrial disease science space. And I want to make sure that we cover that today. We're going to talk about your perspectives of what's happening in the mito world and the convergence of programming that's powered by our donor community that's being done at CHOP and around the globe. But I think first we just need to get to know Dr. Falk a little bit better. We get excited to come in and just kind of understand a little bit about your path to where you got to where you are. And I hope we can start there today, Marni. I'd love for you just to take us back. I always joke with Phil, I got a C+ in biology in high school.
When I first met him, it was a C-.
I thought I got to a B. Phil's got me to a B- at some point here. But Marni, I want you to take us back a little bit on your budding career in science. Take us back.
That's so great. My son is thinking about a career in science now, so we've been revisiting some of this and watching what powers somebody to go in a direction. I think I didn't know anybody in medicine or science as a child, except of course my own doctors. I found out during school that I really like science, biology, physiology, chemistry. And I had the opportunity to do a science project, something way back when that was called the Westinghouse Scientific Program, the Westinghouse Scholarship Program. And so I think you'll laugh. I don't know if we've ever talked about this. I did a project in healing, but it was healing apples.
Oh yeah, tell us about this. I need to hear about healing apples.
Yeah, so it turns out the food industry really worries about health of their produce. But when apples get injured, they have different biologic processes to heal, and it's a big industry to try and figure out how to make apples resistant to injury, puncture injuries or others, so that when they get to the market, people want to eat them and they're not all fungus and contaminated. So I actually spent my, I think, senior year of high school injuring and trying to figure out how to make apples heal or studying wounds. And I was really fascinated about the approach, how you ask a question, how you figure out, the right strain to study and the right methodology. And I wound up winning.
Oh, nice.
Or at least becoming a semifinalist. So that was kind of cool. And then when it was time to pick a degree, I knew I really liked science. I had rotten apples all over the school, and everybody. And I think at that point, I also was a volunteer. Like a candy striper, like classic candy striping, which blew my mind, right? Like that they'd let you into a hospital as a high school kid and let you see things and help. And so when I met with my guidance counselor, she said, "What do you like?" I said, "I really like science and I really like writing." I didn't realize I was talking about being a scientist back then. And she said, "Well, have you thought about medical school?" And I was like, "Well, do I have to think about that now? I'm in high school."
No, this was in high school. This is your high school counselor. Okay.
And my father was with me, and sitting in the meeting, and they said, well, he's an accountant, I should say. And he said, "What do you mean?" And she said, "Well, there's ways to get into medical school from high school, and they lower the tuition." He said, "Tell me more."
Oh, right. That's great.
So it was the beginning. This was the mid-90s. It was the beginning of what they call 7-year medical programs. So in Europe, they accept a lot of kids into medical school from high school, but that wasn't done in America. At first, you go through college for 4 years, and then you apply and go to medical school. So I actually applied to medical school from high school. And so it was a combined 3 years in college and 4 years in medical school. Turns out my husband was in the exact same program. There were only 8 of us, and it was in Washington, D.C., where we wound up going. And it was an unbelievable experience. You can
that is really cool. We should have saved this for the Valentine's Day episode. I didn't realize that. So that's great.
Well, as a matter of fact, it was the second year the program ever existed, and my roommate wound up marrying the other person. So 4 of us actually got married out of this thing.
Oh, how cool.
Separately.
So then you get to school, and now you're heading into medical school.
So I knew I was going to medical school. Yeah, but so I could major in anything I wanted, and I, of course, picked biology. And I was in Washington, D.C., so I got to take the most unbelievable classes with people that were literally working at the White House and, on the healthcare reform committee and, famous ethicists. And just, I had, the best time. Then I realized I had time back then, and I was right down the road from the NIH. So I applied on postcards to every lab at the NIH or every center. And one person wrote back and they offered me a position, and that was the NIH Clinical Center. So I got to spend 2 summers working with Dr. Fauci and amazing people that I just happened to write a postcard, and it just happened to get accepted, and working on new ways to freeze blood cells, which is pretty amazing. And somebody showed me a brand new machine they had to do something called PCR, which was pretty cool, because when I got into college, they were hiring college kids to literally read gels and find the sequence on these giant gels. And now there's a machine that could do it, right? So, I mean, I don't feel that old, but looking back, I feel really old, right? It was like the first PCR machine at the NIH, right? I mean, so it was pretty amazing. And then I think, got exposed to some amazing people thinking so big, right? And really merging research with clinical care. And it blew my mind because compared to what I was seeing in medical school, which is very, introductory, to seeing the people coming up with the cures for the most rare and complex diseases, I was totally fascinated. And I would get to round with Dr. Fauci. I actually got to attend the clinic that they created, which was nothing like what I was seeing in the clinical world of the hospital I was in. But in the research side, they'd created a clinic where they brought together the very best specialists from many different disciplines: psychiatry, neurology, GI, gastroenterology, infectious and allergy, Dr. Fauci. And you know who they were taking care of? They were taking care of all the homeless people who had gotten diagnosed with AIDS, who just by giving blood, they were diagnosed at the Red Cross. So these people who had nothing were given the best care I'd ever seen in my whole life. And it made a huge difference. And that's where I learned that a multidisciplinary team is an amazing thing for patients, right? Because if you have HIV, you have so many problems, right?
Right.
Social and medical, and how do you address them all? And so to see them actually address it all, it was something I took with me forever. And that's really when I came into my own career, I think what stuck with me and what I emulated. I actually had convinced my institution that it was so general what they were doing, of course, HIV care, right? That I should do my primary care rotation there. So I spent 6 months going to this clinic and really learning. And it really just blew me away that's what patients need. They need the doctors to talk.
It's fantastic. I want to come back to that as it relates to today at Children's Hospital of Philadelphia. Talk to us a little bit about, quench my curiosity on kind of where the mitochondria and mitochondrial disease started to kind of rear in your career path. I want to hear a little bit like how that happens in medical school, how it happened through, right out of school. Talk to me about when you were introduced to the mitochondria.
It's fascinating. So I wasn't.
Right. This was such a softball question for you, Marni. Yes.
Yeah.
I did this on purpose.
Yeah. So I graduated medical school in the year 2000. And I had told the dean, who was an obstetrician at the time, delivers babies and gynecology, that I was going into genetics. And he said, "Oh, what do they do?"
Wow.
Right. So the genetics specialty was only formed around 93. So you're not talking that this is a whole, a huge history. There were people that were just grandfathered in. And then, so this is like 6-7 years later that there was a track where you could actually train in bringing genetics to patients. Right. And so again, this isn't that long ago. And when I decided I liked genetics, it was because I really like kids. I loved working with kids. I had a little sibling through a big brother, big sister program who had cancer. And we spent a lot of time together, in this program. And I knew I wanted to work with children, thought I wanted to work in hematology because I had spent so much time doing that. But one day I was in the neonatal intensive care unit at DC Children's, which was affiliated with the George Washington University where I trained in medical school. And I was in the NICU and nobody knew what any of these babies had. They were so sick, right? And they had variations in how they were formed and certain organs not functioning or not formed properly. And they would just manage it. And then one day a doctor walked in. Turns out she was the geneticist. And she picked up a baby and she started rocking the baby and talking to the baby and telling the family what a beautiful baby this was. And that here's what we're going to do. And I was like, who is this magician? Like all of a sudden, like somebody sees the person behind the disease, which I thought was amazing, and how to put it all together. So I actually wanted to train in genetics. I wanted to learn how to do this, right? Like, how do you actually know what caused something and how to help them? To me, that was amazing. And there was an opportunity. I spent a month up in Maine at Portland Medical Center. It was the month before I got married. And there was only 2 rotation choices. And one of them was genetics. So I went and they had one geneticist for the whole state. And so I got there and he says, "You can do whatever you want." He was so busy. So I got amazing exposure. And his name was Dr. David Whiteman, a very well-known metabolic doctor who really was a great mentor for me and exposed me to how you take care of patients with mitochondrial disease and really complex things that they didn't have the tools to solve, right? But how do you take care of people, right? Not just patients, how you really start to understand what his office was above a supermarket and they'd come from all over Maine to see him and he was the only one and he was just absolutely fabulous. And he said to me, "Marni, there's a program starting out in Cleveland, a combined program where you can train in genetics and pediatrics." So I went to look and I was totally taken by the fact that they had an indoor atrium with palm trees. And
that's on the brochure. I like it.
Yeah, they had solved the winter problem and this place was,
too funny.
So I was totally bought, but I bought into it. But it was an amazing place. And that's where I was exposed to people that I didn't know I was going to be exposed to. Legends like Chuck Hoppel and Doug Kerr and Art Zinn. And these became my mentors. And so we would go out and the first patient, when I knew this is what I wanted to do, it was my first year on genetics. I'd finished a year of rotating on pediatrics and seeing, the asthma patients and the cancer patients and the endocrine patients and all sorts of things. And then I was on genetics. And with Dr. Zinn, we drove out to a small town in Ohio called Youngstown, Ohio. And there was a children's hospital named Todd's Children's Hospital. And we would go around in their intensive care unit. And there was a little girl, I remember her name. And I'm not that good at names, as Phil knows. So I remember her. And we were talking to her mother, and she was watching us. Okay. I didn't know what she had. And then the doctor sat down with the mother. She was a Hispanic woman whose English was okay. And he started drawing these Roman numerals, 1, 2, 3, 4, and then oxygen, and then ATP. So he was drawing for her the electron transport chain. And he put an X through 4, and he said, that's her problem. And the mother said, why? He said, oh, we don't know. And I said, what do you mean you don't know? Right? Like the mother said, what do you mean you don't know? And I'm asking, like, what do you mean you don't know? Like, why? Why is there a problem in 4? Like, what's causing that? And the little girl's watching you, but she couldn't breathe because she had Lee syndrome. And so she had stroked. And I'm like, what is this? How could you have a little person watching you who can't breathe on their own? Like, what happened? She used to, and now she was 3 or 4 years old, and now she can't. And so I was really struck with how beautiful this child was and how perfect she seemed, and she had been okay. And now she had a 4 problem and she couldn't breathe. And we went back to see her multiple times. And eventually she passed away because there was no way to go home like that. She couldn't breathe because she had a stroke in her brain that prevented her from breathing without her thinking about it. So she needed a tube down her throat. And after time, you get infections and you can't live like that. Now you can, because care has really advanced and there's home ventilators and other options and non-invasive. But back then, there wasn't. And so I became really like just taken. What did she have? How could this be? And during that time, I was exposed to the beginnings of research. I loved research. I'd been at the NIH. I'd never had time to do my own program. And I was following what was happening. And a bunch of worm researchers at Case Western, 2 actually, a married couple, Phil Morgan and Marge Sidensky, had found a worm that was very sensitive to anesthesia. And when they spent years figuring out what that mutation was, it was a part of complex 1. So why would complex 1 change the effects on anesthesia in a worm? It's very unusual. Like, what was going on here? Like, so now there was a way to actually study what happens when you change the genes, and you could study the mitochondrial function. And then there was a new methodology that they had just given the Nobel Prize for to Andy Fire and Craig Mello, that you could knock down any gene you want and look at the effects. It's called RNA interference. Well, now I was like, well, I'll come join your lab, but I got to figure this out. What causes the complexes to not work? And I had enough data in 6 months, to put in 2 grants. I got them both. One was at the Cleveland Clinic. One was my own K award. And around that time, my son was born, and we wound up moving back, and I came to CHOP. But that's how it all started, was that, why would somebody, go from functional to not? Like, what caused that? That just to me seemed like such a mystery. And it seemed like the tools, again, now it's laughing, right? Because the tools were just beginning. It was the very first tool. But there were tools to study the effects of what happens when you lose a gene, like what happens and when the function's out. And I think it occurred to me, there's a lot going on here, and there's probably a lot to learn. So I'd say the first 5 or 6 years, I just really wanted to understand, do all genes do this? Can any gene do this? And it wasn't until years later, maybe 5, 6 years later, I realized, I think you can treat this. That's what we've been working on ever since.
Phil, it's a path we hear from so many in our clinical community, right? When I asked that question, and I knew that was a little bit of a softball when I served up to Marni, but it's a challenge we have in this space of, helping our community kind of better understand the mitochondrial disease. And of course, UMDF put some investment in doing that. But Phil, I know this is something that we hear early and often.
Absolutely. I mean, that's an all-star lineup of mentors.
Right. Yes. For someone who isn't good with names, that was pretty darn good.
These are my people.
Right. Your people.
These are my family.
To have a chance to learn next to these folks, together with them, and clearly they trusted you to, take on your own thoughts and research projects with it. I don't know that you could ask for a better way to come into a field, and we're very fortunate that connection was made that brought you towards mitochondrial disease, because as you know, a lot of progress over the last decades even, but still a lot to understand about mitochondrial biology and particularly pathology.
Yeah, and I should say there's more people in my story, and they're all connected. So I'm not a morning person. For those of you that know me, most geneticists, it's not a morning thing. Like you start, at a proper time. It's not pre-dawn usually, but Chuck Hoppel would do his meetings pre-dawn to review muscle biopsies on everybody around the country. And so I would get up and get there, and who would be there but Samit Parikh, who was training just like I was, and Bruce Cohen. Yeah. And so pretty quickly I started to understand how complicated the medical problems were, how almost like art, the interpretation of the biochemistry was, and how the causes were missing, right? Like what was causing all this? Was it medication? Was it stress? Was it genetics? Like what was it? And so I think we weren't really able to answer those questions for until a few years later. And I'd say now, as you guys started out the podcast, it's never been better, right? We used to be able to diagnose less than 15% of people with a clear cause. Now we're, in some cases, like Lee syndrome, we're up to 80, 90%. We can solve it pretty quickly. And solving quickly used to mean a year. Solving quickly, I've seen cases solved in a week. So that's a big difference.
Yeah, I want to come back to how we are leveraging genomic databases to help solve and discover a little bit later on here. Let's fast forward a little bit to you now serve as executive director of the Frontier program at Children's Hospital of Philadelphia. Talk to our listeners a little bit about what a Frontier program is at CHOP.
That's a great thing. So about 5-6 years ago, our department chair, Dr. Joe St. Jem, realized there was a lot of clinical excellence, but that it could grow even further, and that the institution could wrap around the clinical with the clinical research to really organize the experience so that it was better for patients, organize the ability to do clinical care and clinical research. And so they started a competition. We were the second year of funding. And so it was several $1 million to be able to go from a program that had a multi-year waiting list and 1 or 2 doctors to a couple month waiting list or less. I think, we can fit people in within the month, and a team of doctors. So really being able to expand the ability to take care of people. And so we had about 3 years to hit our milestones. The first person I hired was our program coordinator, who's now our operations director, Colleen Murescu, to help us build. We recruited several of the faculty members, Dr. Amy Goldstein, who's the Pied Piper of mito medicine, I'd like to say. Everybody knows and loves Amy. Rebecca Ganetsky, a biochemical geneticist, was able to join us from the beginning. She had just come onto faculty, who had trained at CHOP with us. And then Zara Zuela Cunningham, a fabulous mitochondriac who had trained with many people, Ingrid Tyne up in Toronto at Sick Kids, Richard Haas and Bob Navio, and had come through the NAMDAC Fellowship, the North American Mito Disease Consortium Fellowship. That's when we got to meet her, and we were lucky to keep her and grow her through philanthropy in part, being able to take somebody junior and allow them to grow their own program. And so we were able to expand our patient population because of this investment from about 200 visits a year. Now we're up to about 1,300 visits a year. We were seeing half of our patients were always traveling to see us because there's not enough mito docs. But now we see patients from 45 states and 15 countries. And we're able to really integrate clinical care and clinical research. And behind it, we have a whole translational and basic research program so that we could, collect samples like cell lines on patients. Now we can do types of cells that you could turn into stem cells, that you could turn into different organ progenitors, that you can test drugs, that you could start to understand, did this variant cause that disease or didn't it? What's running? So you can finish the discovery piece for people and prove things in remaining cells and DNA from people. We've had the privilege of working with people where their children, were lost decades ago, but somebody had a sample, and now we're able to use modern insight to solve the cause. And more importantly, I think that is important, but more important, there's now hope that there's many, many different ways to potentially treat this. And now it's a matching game. What's the right therapy for the right people, and how do we prove it? So that's where we spend the most of our effort. We have 5 active clinical drug trials with pharma. We have 3 natural history studies, several with you, and other foundations, which is huge. We have a lot of pharma companies. I just did a review. There's about 30. There's soon going to be 33 or 35 companies developing therapies that we try very much to help and give them access to us and our knowledge and our patients and our ability to work together. And we've been able to grow further. We've been able to grow an inpatient service. So rather than just being there in clinic when everybody's kind of okay, to be there on a patient's worst day,
right?
And provide care. We have a nurse practitioner who's able to really coordinate complex care. With Dr. Zolkopli, we did a survey. Most patients have 16 major medical problems. Doesn't matter if they're a kid, doesn't matter if they're an adult, it's often progressive. How do you go to a doctor and take care of 16 problems? When I was in medical school, they told you people were allowed to ask for help on 3. And, and maybe when you were leaving, they asked you one more. So you had to change the model for how you take care of patients. They were showing up with charts, measured in half feet, and you can't go to a doctor who has 15 minutes and ask them to start to review it and counsel you and provide therapy and provide trial. So what we were able to do because of Frontier is set up the program the way it needed to be set up. The time is taken that is needed. Preparation is done where records are obtained and reviewed. The information is given to the patients that they need. The clinical testing is ordered and sent, that they need. The care can be organized, multidisciplinary care, like a neurologist and a geneticist might see them, or they might come in and get exercise testing. Another piece of what we've been trying to do, Brian, is develop better tests. It stinks to have to go for surgery to get a diagnosis, right? Like that's not what people love. So we've created ways to do needle biopsies so you don't have to go under surgery. And now we're doing a lot of non-invasive test development. So lab tests, blood-based and urine-based and genetic-based is big. But even beyond that, the mutation, you might know the biochemical deficiency or problems that also occur you need to treat. But how can you just look at somebody and measure how their mitochondria is doing today? We can't. So we do assessments, strength and physical therapy and nutrition and quality of life on everybody all the time. And then on top of it, we have a whole team working on, can you go on an MRI and you measure mito function? The answer is yes, we can. No anesthesia, no pain, no nothing. We're developing sensors so that you could, kind of like the way you measure your blood sugar, we could measure your mito function. So we just got a nice grant. It just started from the Department of Defense, brings together almost 50 people across the institution to work on this, engineers.
Oh, fantastic.
Yeah, and different levels of biochemical expertise and animal model expertise and human subjects expertise. And we're not just limiting it to people with genetic mito dysfunction. We're also looking at people who have mito dysfunction from trauma or from being acutely ill. So people who have a concussion who were fine and now they're not, turns out they have mitochondrial dysfunction for a time. So can we actually monitor that for them and help them? So that's another example where mitochondria are necessary for so many things. And the autonomic problems that occur after a concussion are mainly due to mitochondrial problems. So these are the types of things we're looking at. Also, we know exercise. You started the hour. Right. Exercise is the best thing you could do to activate your mitochondria.
Right.
But a lot of patients can't exercise when they have mitochondrial disease. So it's like a catch-22. The less they exercise, the harder it gets to exercise. And a lot of our tests require them to exercise to see if they can handle exercise. So what do you do with a kid or a baby? Or what do you do with somebody who can't exercise? So now we're testing whether we can do passive exercise. We don't make you move, we move you. We shake the head or we move the legs. And so that's what Dr. Zolkopli and Dr. Nadkarni in our group are doing. So once again, it's the idea of bringing different perspectives together to do new things that will benefit patients, and that bringing the teams together, so that we care for people, not in the intensive care unit necessarily, where there have always been teams, but in the clinic, right? Where you can make things easier. And if I have to send them to another specialist, it's for a specific reason. Please go for this hormone check, or please go for this heart rhythm check. But we can take care of their overall picture because they're a person and it's all related. It's not just one random problem after another.
Yeah, I mean, clearly you are a top performer when it comes to celebrating teamwork, right? I think about the first time I had the chance to tour your lab and to meet with you in person, Marni, and you make the effort to make sure that I'm meeting every single one of those team members, and how important that is to you. It's just something I've always admired in you. When we get on phone calls, Marni brings a team with her when we get on the phone calls. It's all about collaboration. We should come back and talk about just general thoughts and collaboration too here in a little bit. Phil, it reminds me too, I think about team, I think about the good work happening at CHOP. We'll get into some of the specific projects, but from most recently, one of our accelerator prize finalists from our grant program, where we aim to fund promising research from postdoctoral students, comes from Children's Hospital of Philadelphia too. I know you've had the chance to work alongside moving this program forward?
No, absolutely. Right through our UMDF grant program, we try to solicit the best science from around the world, and regardless of where that science is being done, we want to try to advance the research mission of the foundation. We routinely receive quality and quantity from CHOP with regards to research proposals. And this past year was an example with the accelerator program where Lenny Berg was put forward with Marni as his mentor to work on a project that was viewed as one of the top, under peer review, one of the top rated proposals. So we're really excited to be able to have the funding in place to be able to support his work. And, as Marni mentioned, we have a long history of collaborating, working together. We've funded Drs. O'Kipley Cunningham on a couple of occasions, and supporting the projects that Dr. Falk works on. So it's really a relationship that I think is an important one, that we have a shared vision for where we're trying to take this mitochondrial disease community, improve the outcomes for the patients.
Well, speaking of shared vision, certainly a testament to that is our Lee syndrome roadmap project. We've heard Dr. Falk already talk about her interest in Lee syndrome, and of course, UMDF, along with our partners around the globe, our friends at PALS and Mitochondrial and the Mito Foundation, along with our friends at Lilly in the UK, who made this commitment to help advance Lee syndrome, progress Lee syndrome for the Mito community. And of course, Children's Hospital Philadelphia, along with many of the great institutions around the globe, are helping to move this forward. And the two of you have spent a good amount of your time over the past couple of years making sure that we're moving this ball forward. I was hoping we could talk a little bit about what's happening with the Lee syndrome project, particularly around natural histories.
Yeah, I'll tee it up, and Marni, I'd love to hear your thoughts on it too. But this was an important project to demonstrate that patient advocacy groups could collaborate, come together, and direct the advancement of research on a specific form of mitochondrial disease, Lee syndrome in this case. So assembling the experts from around the world was the easy step, right, to advise these patient advocacy groups. And clearly we wanted to have Children's Hospital Philadelphia involved, Amy Goldstein and Marni Falk on this front, and others. But when you start defining priorities that include, we want to capture data on a global basis, this is a case where the specific experience of CHOP really came through and helped that project to advance. So Marni, maybe you can just talk about the role of CHOP as the data coordinating center for this project.
Yeah, absolutely. I think the partnerships and the relationships are huge. And I just want to give a shout out before I jump in that the UMDF has been incredibly pivotal in this field. So what they did in the early days with Chuck Mohan and Kara and the Strip Matter and the early is they brought the community together, and they actually shared the voice of the patient really early on, not just the voice, but their clear request for people to take this seriously, help work on it. They added legitimacy. And that's why people like me went into mitochondrial medicine and not hematology and not cardiology, because we realized there was a big need that the UMDF had articulated or had brought to an area where there was a field, there was a funder, right? There was advocacy. And I think from those early efforts, which were essential, to recognize the disease, advocacy was a big part of what the UMDF has and does. And I don't know how much other people focus on this, but to me, this has always been remarkable. The reason we got the grant we just got, the Department of Defense, is because the UMDF advocated for the Department of Defense to consider mitochondrial disease worthy of funding. If that hadn't been done, we wouldn't have been able to apply. So everything is sort of connected. And back with the natural history study, we started to go to the FDA and we said, how we, meaning the community, like with the UMDF leading us, how can you develop a therapy for a disease that's so many diseases? That's so many ages, that's so many symptoms, that's so many causes. And they started a critical path innovation meeting in 2015. And it became very clear that it was possible, but it was a hard road. And that a lot of families would need to participate. And for companies to develop therapies that could be approved, the FDA was going to require that we understood what normally happened in the disease. What's the normal natural history of the disease? And we all looked at each other and we said, all right. And we realized it may not be glorious, right? It may not be the pivotal trial where you have the cure. But if we don't do this, we're never going to have the cure. And so we have to work together, because in the Alzheimer's community, somebody in a laboratory can develop a drug and somebody else can pick it up because there's an outcome they use for Alzheimer's. But in mitochondrial disease, what is the outcome, right? Like, how do you know if the drug worked? Did the vision get better? Did the hearing get better? Can they run further? Can they walk? Like what? How do you know? So that's the story of how we all became very engaged, I'd say over at least 2 years. And one of my interests is that I'm a data geek. I love data. I don't like to guess. I don't even like expert opinion. I like to see the data, and then we could all be experts and have an opinion, right? And so when I created the Frontier program, one of the first people I hired after Colleen and the doctors was a data integration analyst named George, who's a rock star. And he had created the first election system in Sierra Leone when they gained independence. And I thought, if he can make all the villages in Sierra Leone communicate and vote and elect, he can come here and help us connect all the different data, the genetic data, the medical record data, the outcomes data. And we started building systems where it wasn't manually intensive. It wasn't working harder. It was working smarter. We were pulling out all the data from the medical records. We were pulling in all the data. And that revolutionized our ability to ask questions and to, again, remember people's names and learn which groups of patients and to study them. But it also gave us the capacity to support the UMDF, because it wasn't just that we could do this for our site. Now we could do this across the world. And I think that was another pivotal piece where the Frontier program and the UMDF really converged. And I think all the doctors, wonderful people, really said, okay, if you want to see somebody with Lee syndrome and know if they got better, you have to look at their tone. You have to look at their balance. You need to look at, their quality of life. You need to look at their parents' quality of life. You need to look at their survival. So they were explaining, over 2 years, the exact outcomes that would matter, that we guessed would matter, right? Because it all matters, and what might actually get better, And so once they did that, we have a fabulous clinical research program coordinator, Laura McMullen, who was able to say, okay, and she was able to create one IRB form that everybody could use, one data template that everybody could use, one system. And so we could actually do this. We can create very high quality data that's very low burden for the families, very low burden for the doctors, but very, very important to pull together what does a patient look like over a year, over 5 years. And under Phil's guidance and the PAGs, the patient advocacy groups from Italy and Australia and the UK, and I'm sure I'm missing some, you came together. And because you came together, I think the world's come together. And so that's a big, big deal. And so when we opened, we asked every patient just as they came in if they'd like to enroll in the Lee Syndrome Natural History Study, and we had nobody say no. So even though they have to complete surveys, even though they have to complete outcomes, everybody said yes. And so we were able to enroll the first 30 people we asked, which is amazing. And so our goal, again, over time is to be able to grow this and enroll everybody, because I think everybody's data is important. But the idea is we have to do things that are cost efficient. We have to do things that everybody agrees on. We have to do things that are palatable to families. We're not doing spinal taps, right? We're doing, how's your balance? How's your, we're asking questions and we're doing some assessments over the computer, for example. So I think it's been amazing to be a part of. I think it's been tricky because we have another effort that the UMDF has supported us on where we curate all the genes that cause Lee syndrome, and we're up to 111. So there's 111 different genes. So if you have a clinic full of Lee syndrome patients, everyone there may have a different gene disorder.
That's 111 within Lee syndrome.
There's 111 genes that cause Lee syndrome. There's probably over 400 now that cause mitochondrial disease. Probably hundreds more. We're still curating. We edited a compendium, the mito disease genes compendium, just to start counting and to make things real, right? If it's in a book, it's got to be real, right?
Right.
So we've got 1 page per gene, and the whole community, about a dozen of my colleagues from around the world and from CHOP helped, adopt a gene and summarize it. And we use informatics to make this more effective and streamlined. About over 250 genes made the first cut. When you read it through, it's fascinating. I learn every time. Oh, there's a gene that just causes anemia, low red blood cells. There are genes that just cause muscle. There's genes that cause everything. There's a gene that can cause every type of named syndrome ever discovered. In some people, it's Lee syndrome. In some people, it's MELAS. In some people, it's MRF. Same gene, same variant. So mitochondrial disease is absolutely the definition of hypervariable. Many different causes, many different effects. And so the fact that we can come together and come up with a way to capture everybody, it goes back to the multidisciplinary, right? Like, how can you provide care? Because we want to help everybody. There's never been one patient that we don't want to treat. We want to treat everybody. And I do think there's going to be some therapies that are specific for this type of gene or this type of variant. But there's going to be many others that are in the arsenal that are, if you have mitochondrial dysfunction, this could help you, regardless of why. And now I think the community is in the matching phase. I love to say that I didn't invent this, but I was told by a chemist, a medicinal chemist, the 20th century was the age of drug discovery. They found billions of drugs, and Phil is an expert in this, so he could tell us more. He literally is trained in this. And then the 21st century is figuring out where they go. Like who, which compound, when. And so, for example, the study that Dr. Berg is doing is to actually take every approved drug from the FDA, the Food and Drug Administration, and some natural products, which mito families are very interested in, and to screen them and say, which of these can prevent stroke in a Lee syndrome model, right? And so I think that's, again, it just shows sort of like the collaboration and the connectivity.
So it dovetails into the MSeqDR project. And Phil, at the risk of me butchering the full name of MSeqDR, why don't you tell our listeners a little bit about MSeqDR? We can open conversation of how this is integrating to what we're doing with MitoShare. And of course, Marni, I know this is a passion project of yours too.
Yeah, absolutely. And Marni should really be the one to provide the details to it. But, fundamentally, you need data to advance research. And that data could be health data, that the data could be metabolomic data, other types of omics, but genomic data is a really important component. It's one of the legs that builds the stool upon which effective biomedical research can take place. So, full credit to Marni and her collaborators that way back in 2012, right? I can still remember this clearly. I was still serving on the board of trustees for UMDF at that time. I had not moved over to staff, and Marni, from coming off in the SMAB, made this impassioned pitch for why we, as a disease community that's predicated on genetics, right, on genomic data, we need to do a better job, right, of collecting that genomic data, bringing it into a central repository, and creating the tools that can allow research to take place and diagnoses to be made and aid clinical care. So, Marni, hopefully that tees it up for you. But since then, I know it's been a labor of love, and one that we've both wrangled with and trying to bring it to fruition. But it's really exciting now, right, that we're actually seeing data flow into MSeqDR.
It's absolutely incredible. So I remember the conversation too, and we said, you know what? It's 2012. Exome data is here. Copy number variation, SNP data is here, arrays. Genome data is going to come. It's a flood. How are we going to prepare? And we're not lucky enough. Like, I always make fun of the CF Foundation, only because they're wonderful, but they only have one gene. One. Right? And it took hundreds of millions of dollars, and they've developed therapies, and it's fabulous and an inspiration. But they have one gene with lots of variants, thousands of variants. We have hundreds of genes with limitless variants, multiple genomes. And the first problem we had is that everybody threw away all the data that was coming out on the mitochondrial DNA. So we had a whole gene, a whole chromosome people were just throwing in the garbage. So that was one point of concern of how they were missing the diagnosis. Because until recently, the big genomic data resources literally didn't even look. It's like, we'll just get rid of that. and then they'd look at the rest. It's a chromosome outside the nucleus. It's in the mitochondria, and you don't have one copy, you have many. So we had to come up with ways for the community to think about that. And all of this became possible from those early days because the UMDF said, okay, we'll support you. And NAMDAC gave us a pilot fund in the beginning too, to say, okay, well, what do you want to build? And so we worked on it, and there's been continuous funding ever since from the UMDF. So it really is a UMDF essential program. And I think has put the UMDF way further ahead than almost any other disease organization because the community is organized around genetic information now, right? And so what happened is that we launched a website, not for families, but for actually the doctors and scientists to have reliable, legitimate information. Not that it's a typo or a sequencing error, that is or isn't a real mutation. And so we started to put together information that doctors and scientists could use. And as a matter of fact, every laboratory that started developing genetic tests would go to our website and look up, well, how do I know this variant is or isn't the disease cause before they put it in their report? And we worked with Dr. Wallace and Marie Lott, who created MitoMap, which is another major resource that we integrated with. We started working with the international community. Phil probably remembers, like, how do you call a variant? How do you transfer a data file? So very basic problems that 10 years ago there was no answer for, that everybody's reached consensus on now. And then we started bringing together all the people behind the scenes, all the lab people and the scientists and the doctors, and we started meeting once a month. And we've been meeting once a month for 10 years now, and to share resources and updates. And to put, information and tools. People were dedicating their sabbaticals to volunteering to create new ways. For example, to look at all the types of mitochondrial DNA deletions that might cause Kern-Sayer or CPEO, chronic progressive external ophthalmoplegia. Lori Kaguni's database, which is to look at whether any two variants in Pol Gamma do or don't cause Alport syndrome. It goes on and on. So much information, but a central hub. And one of our dreams, and I'd say Phil and I collectively, I think this is one of our longest dreams to bring to fruition, but it feels so great, is how do you let the families now interact so they can put their genomic data in so that they're not walking around a conference with a disc saying, can somebody solve my case? Can somebody look at my data? Because of course you can't look at ones and zeros and random numbers. You need a tool and an information system. And so we've now, because of Dr. Zhao Gai, who's my co-conspirator in running this program, and the expertise there that you've been supporting, we have a way to put the data into a place where you can Google it, so to speak, and you can solve it. The same way you would go in and find your directions to dinner, right? We can go in and say, if you speak genetics language, right? Oh, that's a recessive disease. Oh, that's rare. Oh, we've seen that before, and get within minutes all the information. And that's kind of what we wanted. We wanted to turn doctors into bioinformaticians who, if you've ever known anybody who's computer savvy, they don't own a pen, right? You ask them a question, they type something, and they give you the answer. Yeah. You ask a doctor something, they pull out a napkin, they write something down, they shove it in their pocket, they lose it, right? Like it's not informatically equivalent. And so MCDR has turned doctors into bioinformaticians, and it's elevated the rigor. And we were able to leverage that and get an NIH grant. We're the only programs we funded twice to say, okay, let's bring all the experts together and review all the genes that cause mito disease and decide if there's enough evidence to say that and what the level of evidence is. And that goes into a database called ClinGen, which is a precision FDA approved database. And those types of information that we do as experts, which now involves 50 people from around the world that get together twice a month or more to review every type of variant and every type of gene. There's laboratories that participate. There's volunteers. And it's all run by Elizabeth McCormick, our lead study coordinator. She coordinates all of this. There's 10 curators and there's experts. And it's just remarkable because this is the information that will go down the road to whether a gene or a variant can be corrected by gene therapy. Is it disease causing or isn't it? Can it be called out on a diagnostic lab test or can't it? So the community really has organized around MCDR. All the information has been there. And our newest achievement that's just about to launch, two, I guess. One is that families can enter the MitoShare registry and ask to put their genomic data in, and then they're given back a code, and they can share that code with anybody they want. It's their choice. And then their doctor or researcher can go in and analyze their data. So that's huge. And the other thing we've done is all this expert knowledge and correct information and ways to find information, not just genetic information, but like what features are there of this disease? There's hundreds of named syndromes. What is this, heart problems or eye problems? And so we've taken all that information and it feeds now, or it will soon, directly into ClinGen. So anybody around the world working on the laboratory side can pull in the information from MCDR. So it's actually being pulled in automatically by computer. So I think, again, everything is a virtuous cycle, and we start off because we see it, but we don't know how far it'll go. But the fact that we're at a point now where the data is organized, it's a resource that's considered legitimate and like informative and useful. Again, we made a book off of it so that training doctors can look it up and find the information. And then now I think the last frontier is letting patients put their data in so they can share it. And that just launched, Phil, right?
Yeah, Phil.
Yeah, it did. Just within the last month or so, we activated MCDR within MitoShare. So all participants have the opportunity to identify themselves as interested in the program, respond to a couple of eligibility questions, which is really as simple as have you had genetic testing? And from there, it really goes over to the colleagues at CHOP to then begin the process of helping the patient to get the data that belongs to them. They have every right to their raw genomic data. So I love this as a patient advocate, as it's patient enabling, right? These are data that the patient should have about their own healthcare condition or their loved ones. So,
but it's in a useful format for them. It's not the raw data. And
right.
to say that we don't make them call the lab, we have them sign, and then everything happens in the cloud and the data transfers and it goes into a system where, again, it's almost like putting MapQuest into Google. Now you can find it. Right. And so now they can give it back. And so they could be sitting in their doctor's office and they may want to look up a different piece of genetic information. Do they metabolize a drug differently? And now they can. It's all sitting right there for them to look at.
Yeah, it's a wonderful example of how collaboration can, move projects forward. And, I think, Marni, if anything, is being modest about those early days of 2012 to 2015. I sat in on those early calls, 100 people right on the phone, all trying to hammer out what exactly are we trying to do here. I've been involved in projects like that. That can easily just lead to throw the hands up. Say, this is impossible. We're never going to agree on it. Marni would not accept that. And I think full credit to you and Jia Wu for persevering through those challenging early days to where you could get your footing underneath you. And hopefully I was advocating a little bit within UMDF that we have to continue to support this. It's going to take time. It's going to be complicated, but this is important. And now's the payoff. And I really can't be more excited to continue to partner with you to grow this initiative and make it even more valuable.
And it's a true partnership. And even, the ultimate goal is treatment, right? I always say nobody wants to be described. They want to be cured, right? Like they want to be helped. They want to get better. This data is very important in drug discovery, right? If like there's a drug that we give and how much we give, like for example, dichloroacetate, which is a drug some people take with certain types of mito disease. We know that some people metabolize it differently, and we can predict that based upon work of Dr. Stackpole and others, based upon their genetic variants. And so if we can just look at that data, I could know immediately what dose to start them on. Whereas if I didn't know that data and I had to order it from a clinical lab, they couldn't start the drug for months, or I might just start them and it might be too much and they might get sick. So having genetic information is becoming increasingly more actionable. And it's also pivotal for clinical trial enrollment. If you can just say to the clinical trial, oh yeah, here's the link, go type in, you can see my data, and it's right there, they're in.
Right. Certainly allowing us to go faster, right? We think a lot about that every day at UMDF. And I think, Marni is go fast personified for us. And Marni, I think we could probably have a week-long podcast with you, but we probably shouldn't because you've got really important work that you're doing. Clearly, we're grateful for the time that you gave us, but I'll give you the last word. Is there anything else that you'd like to share with the community today?
I think just a huge thank you. I think part of what's been so satisfying is the people I get to work with. I think and that includes you very much. I think the UMDF has always been a big part of my life. We've even allowed my son to volunteer. And I think now it's a big part of my kid's life. And they wear t-shirts that say energy, and they know about energy. And my daughter just had a test with mitochondria on it, and she passed.
Oh, good. What a story that would have made, Marni.
I was like, you knew it, right? And she was like, yep. But I think from the beginning, the reason I do what I do is because, one, it's like the most basic conserved part of us, right? Is our ability to make energy, right? When at the end of our lives, right? We don't disappear. We just stop making energy, right? And so I think that's so pivotal. You know, the fact that you've organized people to think about it and research it and advocate for it and have legitimacy for it, because it's so complicated, it's so essential, and you can't see it. So the ability to create better diagnostics, the ability to create clear, definitive genetic answers for people, that it's not in your head and it's real, right? And now there's things you can do about it, and that you communicate the actionability, right? That it's not hopeless, and that you organize I think you have a program coming up this week about how to understand trials and get into them. I think that's incredibly important. And I think my inspiration is actually you and the families, right? Like, so if there wasn't a need, we wouldn't go to work. But I think it's a partnership, and I don't think of mitochondrial medicine as something you do for somebody. I think it's something you do with people. And I think that's what the UMDF has always brought for me. You know, we're doing it together, right? I don't know how to do what you do, right? And you allow us to do what we do better. And you partner with us, you enable us. Again, I'm grateful to you, and also the families. You know, we've had families, you know, quit their job and start programs and fundraise and, you know, call attention to an area that was in the dark, that now there's models for, and now there's therapeutic leads for, and it's because somebody raised their voice. So I think just the advocacy piece at all levels, from the families all the way up to the integrated organization, is so huge. And I just want to see you successful for decades to come, because I think, God willing, you know, the problem will be which drugs to get people. Right. You know, they'll have so many choices, and how do you select them should be our biggest problem.
Absolutely.
So well put. We are delighted to be your partner along the way, Marni, and thank you for all you do to not just advance the science you are, but elevate the voice. We talk about that so much on this podcast, and when we hear it from our friends in the clinical and the research community, it's that much more important for you all to be saying that. So we're glad that you have that perspective as well too.
Now, I think mitochondrial disease is not just, for anyone out there, not just identifiable. I think it's going to be treatable and curable. It's just we have to keep trying, right? Don't give up, because I think it's about, there's a lot of options and there's a lot of roads to get down. But if we work together, I think we can get there. I'm convinced.
Wonderful. Thank you so much, Marni, for joining us today on the Powerhouse.
Thank you for having me.
All right, Phil. You know, Marni is certainly no stranger to UMDF, right? We didn't dive into that a whole lot, but her service to UMDF, right? There's a service to the mitochondrial disease community where we want her spending her time. But I want to make sure we're recognizing multiple stints on our scientific and medical advisory board.
We ask a lot.
A speed dial to Marni's phone when we've got questions, when we have ideas, right? Marni always strikes me as, you know, the brand I hope UMDF is yes, and now what? What next, right? It's always yes, and now what can we do? Yeah,
there's much to admire about someone like Marni, and we heard about her path and how she got to it, but you know, the passion, you know, the way in which she conducts herself, and that, you know, she's really trying to make a difference in terms of what's best for the patients and everything she does. And I know I'm personally really fortunate to have a chance to work so closely with someone like that, but our community, I think, is the broader message, is really fortunate to have someone like a Marni Falk involved.
Yeah, I think we all need to recognize that attitude of, yes, this is tough, right? There are tough things for us to figure out. And when she was given that cystic fibrosis example, like, wow, that was a real moment for me to think that through, the difference between the one gene and the hundreds of genes and the thousands of variants in there. But it's always buttressed with so let's go work harder. Let's go figure this out.
acknowledge the challenges, but then
Right.
set about trying to address them and move the field forward. And
Yes.
that's what it takes, right? Like
Yes.
a relentless positivity that we will do this. And I know that helps fuel me and what I do here as part of the foundation. And hopefully it translates to our patient community. It provides that message of hope that we know this is hard. But a lot of really smart people are working on this, and there's exciting times ahead because we're making real progress right on the research. And a lot of that is due to very talented people like Dr. Falk.
Yeah, it's a good thing when it all starts coming together, right?
Yeah.
Despite the fact that there's mountains behind mountains, right? That's the Haitian proverb I have right across from Demon Gen Man. That's how I think about it every day here. There's going to be a mountain behind that mountain, and we're going to climb that mountain, right? We're going to keep going. It's that positive outlook there. So certainly Marni's a shining sample of that. And of course, as we kicked off the beginning of this show, that's what makes this whole community special. Like we're all thinking about something that's bigger than ourselves, something that we can do to help move this forward. And I just, again, as I reflect on this time of year end is always a time for me to say, like, what else can I be doing for 2023? Like, what can I do to harness that type of energy? So one thing everybody can do is check out this show on our YouTube channel where you can find all the Powerhouse shows. Be sure to follow us on social media. That's not a shameless plug to get likes on social media. It's an important piece of what we do to talk about these programs. We want to make sure we're showcasing the impact of our community giving, showcase the impact of the bright minds like Dr. Marni Falk who are putting these things together. So check us out on umdf.org, on Facebook, on Instagram, and LinkedIn as well too. And until next time, everyone, please go fast.